mite biology and management. We have pest management in greenhouses, and pest management really it focuses on decreasing those pests as much as possible, trying to find the most effective way to decrease those pests economically, environmentally, and efficiently. And I just wanted to clarify something. When I say pest management, that's not specific to just insects and mites, but it's the whole scope. So pest can be anything from weeds to insects and mites to diseases, bacteria, viruses, everything that affects the plant in an unwanted way. So why are insects and mites attracted to greenhouses in the first place? Well, there's a ton of food for them, which is a great start. It's a good climate, especially when it's kind of nasty out, just like we have today when it's raining slash snowing and freezing. These insects and mites need somewhere to keep warm and protected so they could reproduce and continue their life cycle. And also, there's not too many enemies in here, which is really convenient for these insects and mites because in the outside world, they're not sheltered. So they are susceptible to all of these extra enemies that are going to make it harder for them to survive. And then here we have again that integrated pest management. This is just an overview for everyone who has already taken the integrated pest management course, but for the people that are new to this program and new to this term, let me know if you need any further descriptions of this. But the goal of integrated pest management is to mitigate pest damage while protecting human health, environment, and economic viability. And the integration of tactics include biological, which is using organisms, chemical, and the chemical is based on the products, and cultural is more abiotic end of it, manipulating the climate, using different, mulches and whatnot, but we'll get into the cultural control practices, and then physical and mechanical, which is actually removing these different insects out of the greenhouses by hand. And the components of an IPM program is pest prevention, pest identification. And I want to stress this pest identification because if you do not know 100% with identification of your insect, disease, whatever you might be wanting to control, if you don't know that specific identification, you cannot accurately find a product to decrease this. Of course, you can use cultural control mechanisms, if that's fine, because it's just manipulating the environment, adding different nutrients, providing the correct components so the plant will not be stressed out and it will be completely healthy. But when you're dealing with products, you need to 100% know this identification because the product that you use will depend on what the identification of the insect or pest that you're dealing with is. And if you have the incorrect pest identification, it may not work on that specific pest that you want to get rid of. And then it's just a waste of money. And you can get into legally some legal binds. So I would definitely confirm that you know the identification of that specific pest. Monitoring and population assessment. And a lot of these greenhouses and plant propagating facilities, they'll have someone designated to scout and monitor the populations of different insects and mites using different sticky traps, different trapping mechanisms to have an idea of what they're dealing with and be able to spray accordingly. Control action guidelines, integration of tactics. We explain the biological, chemical, and cultural, and then physical and mechanical. So preventing this, you want to make sure they're kept out completely, inspect incoming stock, and then isolate the stock for at least a week until actually integrating the stock into the current stock that was already in the greenhouse. Mechanical exclusion, weed removal. You want to make sure all the weeds inside and out of the greenhouse is completely eliminated. No extra flowering ornamentals either. and then prevent year-to-year carryover and remove the residues and sanitize every single year and every season. So we discussed, why pest identification, insect identification is so important. If you misidentify something, it can contribute to a failure and incorrect biological or chemical controls. And this may actually, increase costs for you and increase the pest populations as well. So the common greenhouse pests that we're going to be going over, common greenhouse insects and mites, are mites, thrips, whiteflies, aphids, fungus gnats, short flies, and mealybugs. And then we also have scales on there. The characteristics are very small, hidden, short life cycle. So with a short life cycle, you're going to have an abundance of different insects that are coming in because it's so short, and they can reproduce faster. And the numbers build fast because of that short life cycle. And here's a pretty neat diagram just to show you the size differences among those common insect and mites that are common within the greenhouse. So you can see the mealybug is the biggest, with the fungus gnat being relatively smaller, and then the short fly, the whiteflies, and the aphids relatively in the same size, and then the thrips and mites being the smallest size. So you can get relatively a good idea of what you're dealing with and the size of these insects and mites compared to others in the greenhouse. So for mites, a lot of times we have a spider mite issues, and you can see spider mite issues by identifying the web-like structures that are within the foliage. And then you can see these tiny little creatures crawling. They have 8 legs total, and they look as if you're seeing it right there. Sometimes they leave their exoskeleton behind, and their exoskeleton is very tiny bodies that are clear. And here's just an example. As you can see, it's like that web-like material that's around the foliage, and you can see specks that are stuck in there as well. And those might be eggs. Those might be the spider mites that have already died. The skeletons of them, or the frass, the poop that they leave behind. Under the scope, this is what you're going to be seeing. So those larger orange figures with the dark orange spots on their upper bodies, those are the adults. And then those circular objects, the smaller ones that are light orange to yellow and clear, cream-like, those are the eggs. And then you can see like white specks throughout also, like the larger white specks. And those are the dead spider mites that are left behind. And then you can also see the black specks, and that's the frass or the poop from the spider mites. Spider mites also have no wings, and this is not an insect. As you can see from the beginning, I kept on referring to them as common greenhouse insects and mites because mites are not insects. With this, the 2-spotted spider mite has 2 different spots on the left and side of its body. And the other species include Lewis broad and cyclamen. Spider mites attack 300 different species of plants, and their life cycle is in roughly 8 days. The females lay 100 eggs, 100 or more eggs per life cycle. So as you can see, they are producing a lot of eggs. On top of that, it's a short life cycle, so the reproduction rate is going to be very fast. They are sap beaters. So what they do is they stick and suck the nutrients, the plant nutrients, out of the phloem, so that they really want to try that nice, juicy, sugary substance that the plant produces. And they are sucking these nutrients. And as they are sucking these nutrients, what is left behind is that loss of chlorophyll for the plant. And since that plant has a loss of nutrients in those specific spots, those spots turn to a yellowing. And that's also called stippling. So that stippling is just yellow dots caused from sucking insects or sucking insects and mites. They also are going to go under leaves around, specifically under the undersides of leaves, not the top of the leaves, around buds because a lot of nutrients are there. And they prefer tender leaves, you know, that new growth, because a lot of their nutrients are in the new growth and it's easier to penetrate that cuticle layer. So mites monitoring, you can scout for these. You can be using sticky traps. The sticky traps are just these square traps that are sticky on both sides that you put in various spots throughout the greenhouse. And from there, you just check them once a day to count how many insects that you find. Maybe they're all spider mites, maybe they're thrips, maybe it's none. So just from taking these random samples, you can have a good idea of what kind of pest populations that you are dealing with within your greenhouse. And what you can also do is visually scout, and you're looking for stippling or mottling of the leaves. And that mottling of the leaves is very similar to the bacteria because of the mottling in the mosaic. And that is just the discoloration of the leaves and having yellowish irregular spots. I showed you guys in a couple slides of the webbing that occurs here, but you are going to have severe webbing. It's going to be very similar to a spider's web. That's where they get the spider mite name. And then we also have premature leaf drop. If you guys are not familiar with premature leaf drop, it's when leaves drop as they would, you know, right now all the leaves are dropping in fall because that's the right time because it's going into dormancy. Premature leaf drop is when the leaves drop prematurely. So if you see leaves dropping in July, that is an issue. You can see that there's some underlying factor that's playing a role in why the trees are dropping its leaves so early on in the year and not when it's supposed to. And then also you want to examine under the leaves because that's usually where the spider mites feed on. So you want to be looking for the eggs. You want to be looking for the adult spider mites. And you also want to be looking for the dead skeletons that are left behind. So cultural control over mites, you can reduce plant stress by making sure the nutrition level is optimal, and you want to just reduce plant stress as best as possible, making sure you're irrigating, not overwatering, not underwatering, mulching if appropriate, making sure that the pot's in the correct size of container, making sure the air circulation is optimal. And you also want to make sure no other pest issues are occurring, if that's diseases, bacterial, viruses, fungus, whatever it might be, making sure those are also under control. Thrips are tiny, yellowish, and they're very linear and long. And the most common that you're going to run across is that western flower thrip. Thrips cause deformed leaves, decaying flowers, silvery flake scars, and that black fecal spots, or the frost that's left behind. Thrips are also sucking insects, similar to the spider mites, how they suck those nutrients, or the sap suckers. So they pierce plant cells and they are feeding on that sugary substance from the phloem. They focus on young growth and the flowers specifically. And they also have 150 to 300 eggs that are laid by the females in each life cycle, which is roughly 12 days long. So the spider mites reproduce quicker, but the thrips are reproducing at a higher rate when it comes to eggs, even though it's a couple of days longer in their life cycle. You also have to watch out for thrips because they are vectors to various different viruses. So you definitely want to watch out for those. If you have thrips, other issues can come along with the viruses, and the viruses are the big ones that we want to watch out for. Like I said, viruses, you cannot treat them. Once you get it, I mean, you have to deal with it. You have to get rid of all of your stock and start pretty much new. But with bacteria and funguses, you can actually treat those. So it's the viruses that are really tricky, and you want to treat those. So when you see those thrips, there might be something more than just an insect problem at this point. It might be at a viral level. And then you can monitor thrips. Early detection is key. Really early detection for anything is key, but especially for thrips, because we need to control this as best as possible so it does not stress out our plants and deform them and stunt them, but also we don't want to have the thrip spread any kind of viruses. So I explained these sticky cards in a couple of slides ago, but for thrips, we want to be using blue or yellow cards. If you guys are not familiar, here's the sticky traps on the right where it says BioWorks. And what happens is this whole entire area is sticky, and they have this graph, which depending on how many insects or how many pests land on this, they can calculate how much is actually going to be in the whole entire greenhouse and get a better representative number in their mind for what they have to treat for, and if they should, take charge and treat the insects or mites now. So for the blue or yellow cards that will be used, a minimum of 3 should be used. 1 should be placed near doors, vents, and then thrip-sensitive plants, depending on the species as well. 1 sticky trap should be used per 2,000 feet, and it's placed just above the canopy. So that means you have 10 tomato plants and they are 12 inches high. You want to place that at the 12-inch high level to make sure that top of the card is in level with the plant itself. Another method that you could do with thrips and mites is you could place, and aphids as well, you can place a sheet of paper underneath the branch, and you just whack the branch 1 time, and from that one whack will fall the different insects, the different thrips, and from that point you count, and depending on how much you count will determine that threshold if you would have to treat or not. Cultural control methods of thrips: remove weeds that may harbor those viruses, and then crop alternation. I haven't mentioned that one yet either. It's going to be very important that you alternate all of your crops. Even if you're in a greenhouse setting, you don't want just one crop, because if you keep on having that one crop, that means the insects that are more prone to that one, or the different pests that are more prone to that one plant for feeding on, that has more of a potential to overwinter. And if it's a disease, if it's a bacteria, if it's an insect, whatever it might be, it has more of a potential to overwinter on that plant species and come back the next year. So crop alternation is going to be a huge method in our cultural control tactic. Aphids have a pear-shaped body and cornicles, or tailpipes, as you see on the right, those black little guys coming out of there. And the most common species are green peach aphid, melon or cotton aphid, and potato aphid. Aphids have a high reproductive rate. They have 100 live young females at one time. They're all asexual as well, so they do not need a male to reproduce. And they have live birth, and their life cycle lasts for 7 to 10 days. So this one has the highest reproductive rate in all of our greenhouse pests, or greenhouse insects and mites. 1, because of their life cycle, how it's 7 to 10 days. 2, it's asexual, so it does not need a male whatsoever to reproduce. So those females can keep on reproducing, and all the females are laying the eggs as well. So instead of having, let's say, half and half female, half male coming out, and just that half of female laying those eggs, now 100% of the eggs that are hatching are female, and they are going to hatch even more, 100 more eggs. And then that life cycle is just going to keep on going over and going over and going over. This is another sapsucker. It's just piercing the stems and the leaves, specifically the leaves here. They really focus like right before the leaves occur on the stem, because that's where a lot of the nutrients are, but they also are going to focus underneath the leaves on flowers, and in a big aspect, they produce honeydew. The honeydew that they produce, they secrete this honey substance, which goes on the plant material. That honey substance is their pee. So from this point, or their pee in their poop. So from this point, they have this honey, this sugary substance on the foliage itself. So black sooty mold, which looks very similar to powdery mildew, but it is black and it covers the whole entire surface of the foliage and stems. The black sooty mold grows on the honeydew. So a lot of times if you see this black sooty mold, you can also indicate that there are aphids present because of the honeydew production. And the black sooty mold decreases photosynthesis and ultimately makes the plant more stressed out because it can't produce those sugars and provide itself food. Here's a photo of that black sooty mold that is on the underside of the picture lower, the one lower to the page, but that is black sooty mold. The plants are also stunted, so decreased growth. It has curling or cupping of the leaves, and you can also see that curling and cupping of the leaves on the lower photo. And then that sticky honeydew that results in sooty mold, black sooty mold growing all over it. And aphids also transmit viruses. So when you see aphids, you might also have another issue with a viral issue, just like thrips. When you monitor for aphids, you want to be using the yellow cards. And you should know that some aphids do have wings, but most aphids do not have wings, so they will be crawling. And whenever you scout, look for those insects on the stems, and specifically for that fresh new growth and under the leaves. And you'll also be looking for that sticky honeydew on the leaf surface or black sooty mold. Many times you can be looking for the adult aphid, as what you see is like the orange organisms in the photo. But if you look at the white little specks, those are the exoskeletons of the dead aphid. Another management practice for aphids is to avoid excess nitrogen fertilization, because whenever you apply a lot of nitrogen, you have that flush new growth. And with that flush new growth, you're going to attract more aphids because they prefer the new growth over the old growth. And all around, it's going to stress the plant out even more by applying that nitrogen, which is going to reduce its resilience towards the aphids and trying to combat the aphid populations and infestations. Whiteflies are going to occur in all of their different stages underneath the leaf surface. Their life cycle is a 30-day life cycle, and they're highly visible. As you can see, they are larger in appearance, and they have wings, and they also have many host plants. The whitefly damage includes yellowing and discoloration of the leaves, stunted plants, that honeydew and mold. So the honeydew that's produced from the whiteflies and that sooty mold coming on to feed on the honeydew. And they also transfer viruses. For monitoring whiteflies, you can be using the yellow sticky cards and then do the plant inspection. Look for the undersides of the leaves. Look for honeydew, sooty mold, and then shake the plant for adults by placing that paper underneath the plant. Give it a nice whack and see if any fall, and that can be a great identification if there is any whiteflies present. Cultural control practices for whitefly management includes using the plant-free periods, and then time depends on temperature for cooler temperatures and keeping plant-free longer. Mealybugs are definitely a lot easier to look for because they're large. They're big, they're about 1/4 inch. They are waxy, cottony organisms. They're also sucking insects, very similar to aphids, thrips that we were dealing with in the past, and they produce honeydew. They're also going to have that black sooty mold as well as an ID key. They stump plants. They're going to decrease the growth because they are sucking those plant nutrients and sucking those sugary substances from the phloem, and that decreases the plant nutrition levels and stresses out the plants. They attack all parts of the plant. They are really not specific to any, so you can be looking at the stems, you can be looking at the leaves, fruit, flowers, whatever it might be, but they are going to be attacking everything. They do prefer that new growth just because it's a lot easier to penetrate that cuticle layer, and it has a lot more nutrients in that area and sugar in that area. But you can be looking throughout the whole entire plant. It takes 30 days to complete their life cycle. And because they're so large and visible, it's going to reduce that marketability for the plants. A lot of times, and I don't recommend this, but with different insects, because they're so small, you can still sell them without having anyone, raise a red flag. With mealybugs, if you have them, people are going to know and they are not going to want that plant material. So it's going to reduce that marketability aspect for the plant material. So monitoring mealybugs, you're going to be scouting for them, looking for white flakes on the midribs, leaf axils, and the undersides of leaves. And you're going to be looking for the honeydew and black sooty mold as well. Fungus gnats are small flies, and they feed on the root systems, the stems, and decaying debris. So, getting rid of all of the decayed plant material and the stuff that you've once thrown out, making sure that you take the trash out and burn it properly is going to be very important. And making sure that soil media is sterile and that you're not bringing in native soil that has not been pasteurized. Fungus gnats need damp media, so trying to make sure that you don't consistently have that wet media for long periods is going to be very important. You still want to have that perfect combination of making sure the media is moist but at the same time not overwatering and not having the root systems dry out either. The life cycle lasts for 30 days. And due to the fungus gnats, this might promote diseases such as like the Pythium, verticillium, and gray mold or botrytis. For fungus gnats, you can be using the yellow cards for the horizontal orientation, and then you can also be cutting up slices of potatoes and placing them on the surface, and from that, the soil surface. And from that point, the fungus gnats will feed on the potato, and you could identify how many are in that area, depending on how many land and are feeding on the potato. Cultural control management for fungus gnats includes irrigating less and removing standing water at any times, and make sure that sanitation is kept up, especially with containing the compost as well, because it will overwinter in the soil. Shore flies are small flies that feed on algae. They must have damp media as well for the shore flies to develop and survive. And they leave frass on the plant material, so that's going to be a sign that there are shore flies present. And they also may move Pythium spores and other fungal spores as well. So a cultural control mechanism is really just eliminating that algae. I know a lot of you on your cuttings and other plants that you've done, now they have that layer of algae that is forming on the soil surface. So you really want to eliminate that algae, because we could be bringing in shore flies and other diseases that are coming in. So trying to eliminate that algae, and that's really just removing that maybe a half inch or a centimeter from the top of the soil media, and just eliminating that. And if it comes back the next week, you could take your cutting or whatever plant material that you have used and put it into another media, into another container as well, because it might have been contaminated. So for greenhouse insecticides and miticides, just make sure that you read the labels very carefully. They might have certain restrictions on them, so you need to read them carefully and determine what kind of pest it is, and then what kind of crop that you're dealing with as well. And then from that point, figure out what kind of product that you would like to use. And you also want to be safe at all times, really looking at that label, looking at the crop safety, and then determining your route from there. And then from that point, determining your active ingredient will be beneficial depending on what you want exactly, and what plant species and what pest it is as well. The imidacloprid is very popular. Marathon or Admire, Safari, Avid, TriStar. Those are all going to be really popular products. That doesn't say that you are going to be using them. That's what's so special about really being educated in this field and going into it if you do end up in a greenhouse or plant propagating job position is you can really do the research on what kind of product that you're going to be using. How how potent is this product? What is the residual? What is going to have long-term effects for the environment? And what is going to be the most effective for that certain pest with the host? So you can be doing your own research, and you are going to be the one that's going to determine what kind of product that you use. And here's just a chart that you can go across. And with the target pest or for product Marathon, you can be using aphids, whiteflies, soft scale, fungus gnats, and leaf miners. For Safari, whiteflies, mealybugs, aphids, soft scale, fungus gnats, leaf miners, armor scale. And that TriStar, whiteflies, mealybugs, aphids, leaf hoppers, thrips, leaf miners, and scale as well. And then you also have to look at the IRAC class and determine what you're going to use specifically. And if you use that application again, that it's a different IRAC class. All of these products are systemic pesticides, and with systemic pesticides, they are taken up throughout the whole entire plant. If depending on where it's applied, if it's applied at the foliage or at the stem or at the soil base, that it's applied throughout the plant that goes down to the root system up until the tip. So all of it is being taken care of, and not just one central area on the plant. Now, these foliar sprays include Distance, which is for whiteflies, scale, fungus gnats, and short flies. Talus, which is for whiteflies, mealybugs, leaf hoppers, scales, and also crawler. And we have the foliar sprays continue with Conserve, or the active ingredient spinosad, for caterpillars, thrips, and leaf miners. And we also have Pylon, which is for spider mites, broad mites, rust mites, beet, armyworms, loopers, thrips, and caterpillars. We have a lot of different miticides that Avid is commonly used, and we also use the hexagon quite frequently. Plant resistance of different pesticides occurs when you keep on using the same exact active ingredient over and over again on the same exact plant. Usually for whenever you're applying pesticides, you want to rotate the active ingredient for the IRAC group so you don't create resistance whatsoever.